So we're announcing a collaboration between AstraZeneca and the Broad Institute to discover new leads against a number of viral and fungal pathogens through screening of their collection of diversity-oriented synthesis compounds and targets that we provide.
The work will address the growing problem of drug-resistant bacteria. And so what has happened over the past 30 or 40 years, bacteria have begun to evolve in a way that allows them to evade the current drugs that are available to treat patients with infections. And so there's a critical need to develop new classes of antibacterial agents as well as antiviral agents. And the key here is, again, new classes of compounds that can take on these bacteria in a way that we haven't been able to in the past.
The DOS, diversity-oriented synthesis collection, that the Broad Institute has built over the last few years covers a chemical space which is non-overlapping with the one that our own files cover. And also the Broad Institute has developed expertise and skills to follow up on those starting points from their libraries. On the other hand, we bring expertise in infectious disease, validated targets, and I think the combination of the two is going to be very powerful in order to start a new program against many of those life-threatening diseases.
AstraZeneca is one of the few companies that has maintained a commitment to discovering new anti-infective agents. We believe their strength in bacterial genomics and biochemistry will facilitate early-stage discovery efforts, and their expertise in process chemistry and human clinical trials will ensure that patients will benefit from any drug that is discovered in the collaboration. What really makes this project possible is the fact that at the Broad Institute we can work on a scale that is compatible with industry. Even big companies couldn't do some of the things that we've been able to do here at the Broad, including creating such a large new type of compound collection that is perfectly suited for this type of collaboration. So it's really our ability at the Broad Institute to work on a scale that is so uncommon in academics to be able to match skills with a large pharmaceutical company.
So DOS is diversity-oriented synthesis. It's an approach in chemistry that aims to maximize the diversity of compounds one produces with the smallest number of similar chemical steps. Very simply, it combines two principles. One is for accessibility, synthetic accessibility, and the other is chemical diversity. Now, chemical diversity, particularly in infectious diseases, is very important. You will know that many of the drugs that we have against bacterial pathogens, for instance, are originally derived from natural products. Natural products have very complex structures that are sometimes not easily accessible synthetically. The diversity-oriented synthesis approach is trying to combine both, and thus gives us chemical diversity and structures that could look like natural products, but can be accessed chemically and optimized in the laboratories.
So what we do is harness modern synthetic organic chemistry to make small chiral fragments. So the word chiral means handedness in Greek. And just as our hands are non-superimposable mirror images, we make our compounds in the same way. Nature does this, but we make all possible combinations in a way that nature doesn't. So we build small chiral fragments and then couple them together to assemble our compounds for screening. And so this brings in features that nature doesn't give us with our compounds, meaning that we know what the structures are, we know we can scale them up, and it's easy to perform medicinal chemistry on those compounds. So those are the features of a diversity-oriented synthesis, or DOS library, that we've built here that is so attractive to a pharmaceutical partner like AstraZeneca, because they are confident that we'll be able to rapidly perform the medicinal chemistry on the exciting hits that we already have for this collaboration.
With infectious disease, by the time you start screening the target for a new starting point, you pretty much know that the target is validated. You can test that in vitro, you can test that in preclinical models. In other disease areas, you carry that risk all the way to phase 3. In infectious disease, we have fewer targets to work from, but they're very valuable. Every one of them counts, and it becomes increasingly difficult to find a good chemical starting point. This is where our collaboration with the Broad Institute will help us broaden the chemical space that we explore against those very valuable, highly validated targets.